Joining us today. My name is Andrew Needles. I'm the product manager at VisualSonics, and today we'll be giving you an interactive webinar looking at the VevoCQ software. And I'm joined today by my colleague Stephen Butters. He's an application specialist here at VisualSonics in Toronto. Stephen will take us through a short presentation outlining some of the applications and workflow for VevoCQ. He'll then take us through an interactive session running the VevoCQ software with some example data sets to hopefully give you a good sense of the typical workflow and tips and tricks for acquisition and data analysis. Just a few notes about the webinar before I hand things over to Stephen. We will be providing a recording of today's session. We'll make that available, and we will email after the session today. Just a reminder that all of the lines will be muted. This will help us eliminate any background noise that's happening out on any of the lines and help us create a clear recording and a clear listening session today. That being said, since we won't be able to hear you over the voice chat, please use the text-based chat window at the bottom right-hand side of your screen. You can use this for asking questions. If you have any technical difficulties, please let us know. We have a team of people here that can assist you. We'll be taking some questions both halfway through the presentation, after the presentation segment, and then after the demonstration segment of today's talk. So we will answer those questions via the chat. And the expected duration is on the order of approximately 30 minutes with a few extra minutes for questions at the end. Just before we get started, I do want to thank you for joining us. It's a great interactive session to have all of you here on the webinar, and we do these fairly frequently, so please go to visualsonics.com/webinars to learn more and see some of the upcoming sessions. So at this point, I will hand things over to Stephen, and he can tell us all about VevoCQ.
All right, great. Thanks, Andrew. Thanks for everybody for joining us. I'll give you an outline of what we're planning on heading through here in this presentation. As Andrew said, we'll do a presentation. We'll take a little pause for any questions that may have come up during that short presentation, and then we'll move into a demonstration of the actual software itself and some of the capabilities. So we'll go through a little bit about the software, about VevoCQ, and about nonlinear contrast imaging in general, and about the Vivo 2100. So for those of you using the system, you'll be familiar with some of these modes, some of these types of images. For those of you not using the system, just to give you an overview of what sorts of things that we're talking about, the sorts of things that the system is capable of doing. In cancer research specifically, and certainly other types of research, there's lots of different imaging modes on the 2100 that are applicable. B mode is the standard basic brightness mode, the standard grayscale ultrasound imaging mode. It gives you a good view of the anatomy that you're particularly interested in real time. Other modes, such as the Doppler modes, are for looking at vasculature and blood flow. We have an example here in the upper right of the slide of a power Doppler, a 3D power Doppler. This shows the vasculature in and around the abdominal aorta, the renal artery. Most of these modes, something to keep in mind is that most of these modes can be applied to pretty well any area that you can see. Essentially, if you can see it, anatomy under B mode, under grayscale, then you can apply modes like power Doppler or nonlinear contrast to those particular pieces of anatomy that you happen to be looking at. When we get into contrast, perfusion contrast modes specifically, there's two types of modes, linear and nonlinear. The example that we have down here on the lower right with that copper-colored overlay is nonlinear contrast. And nonlinear contrast is primarily what we're going to be talking about. It differs substantially from linear contrast in that it's a direct visualization of the signal coming from microbubbles. and is much more sensitive. And VevoCQ works strictly with data from a nonlinear contrast mode. And this allows you to do all sorts of interesting investigations into angiogenesis, vascularity, perfusion kinetics, and things like that. So once you get into VevoCQ, once you take your nonlinear data into VevoCQ, you've got a lot of tools available for really in-depth analysis of things like perfusion kinetics and various other types of metrics to really investigate what's going on in perfusion in the area that you are interested in looking in. So as we've stated, this is an advanced post-processing tool. So VevoCQ is something that you would do on your data after you capture it. So you would go in, you'd image, you'd capture a series of data, and then take those data into CQ and start to look at them. You can draw regions of interest, up to 4 in the area, and you've got an example here of those regions of interest drawn around a tumor here. Once you do the perfusion analysis, you do curve fitting, so the software will take those nonlinear data and fit the signal from the nonlinear contrast to curves, to curve algorithms. It generates parametric images for you, so we've got a little visual example here. We'll have lots more later on. These parametric images are really nice because they're qualitative, so they allow you to really separate out by eye very quickly differences in regions within the tumor, differences between animals, between treatment groups, between days. Those parametric images are tied to hard quantifiable numbers depending on the types of metrics that you're looking at, all of which, as is typical for VisualSonics, are exportable, fully exportable as flat text files, so you can actually get your numbers out if you want to take analysis further and start to drill down even deeper into the data that you have. So the VevoCQ contrast analysis software is, to date, the only analysis software that's designed specifically for small animal research, for nonlinear contrast work, and it's designed strictly for the Vivo 2100. We get questions from time to time about the Vivo 770. The type of data that the 770 produces is not amenable to the type of analysis that VevoCQ does, so it's strictly for the nonlinear contrast data from the Vivo 2100 system. Microbubbles, for anybody who has not had an experience with microbubbles, with ultrasound contrast agents, we refer to them as microbubbles because they really are bubbles. They're around 2 to 3 micron in size bubbles, filled with an inert gas, lipid shell on the outside. And these mimic red blood cells. They're injected into the venous system, typically through a tail vein catheter, but there are other portals and methods for doing the injections. And then in nonlinear contrast, we have 2 transducers that work with the Vivo 2100 in nonlinear contrast, the MS200 or 250. These are our low-frequency transducers, a 15 megahertz center and a 21 megahertz center frequency probes. The nature of nonlinear contrast is that it works better at a slightly lower frequency, so this is the reason behind using those probes. So there's a video clip here that's just buffering for some of you and is playing for others. What you're seeing here is a bolus injection into a mouse, and then the contrast agent flowing into a subcutaneous hind limb tumor. So typically this was done with our typical approach, which is a 50 microliter bolus injected via a tail vein catheter into a mouse, and then you see where the copper-colored area is. That's the nonlinear contrast image. So that is the image with the tissue suppressed, strictly the signal from the contrast agent. The gray on the left and right side is your B-mode image. So in nonlinear contrast, the nonlinear contrast signal, the signal from the bubbles, is being co-registered with the B-mode signal. So this allows you to see anatomically in B-mode where you're looking, what you're looking at, and then looking in the nonlinear contrast mode where you're actually able to see the bubbles flowing in. You can see as the video loops around that the area of interest goes from almost completely blacked out with a little bit of nonlinear signal from the surrounding tissue near the skin line to quite lit up as the bubbles flow and perfuse into the area of interest. So typically going into VevoCQ, this is the same image here. So again, we have the nonlinear contrast image on the right, beam mode image on the left, co-registered together. We draw on 4 regions of interest in and around the tumor. And then this is typical to the curves that you get from VevoCQ, where the system has looked at the signal, the nonlinear contrast signal, and has plotted the wash-in, the reaching of the signal up here at a peak level, and then the signal tailing off to a sort of a constant level of circulation. And you can see the different levels of signal for the different regions of interest. The total tumor here, this purple region up here where the tumor is very highly perfused. So drawing different regions of interest in different places within tumors or within tissues that you're looking at is very interesting, because you can see quite clear differences in the types of data that you're getting from those areas. Moving forward from just the curve fitting, we have some examples here now of some of the metrics that are produced by VevoCQ. We have peak enhancement, time to peak, and wash-in rate. This is just a small subset. There's a wide range of data that are produced by CQ, rise time, wash-in area under the curve, and this also depends on the model that you're using to whether it's a bolus injection or a destruction reperfusion. That'll become a little bit more clear later on when I actually show the software. I'll show a couple of different examples. We have some examples down here at the bottom of the parametric images that are produced here. Again, peak enhancement, time to peak here, and wash-in rate here corresponding. So you can see visually there are some very clear differences here, say in the time to peak from the other two. The wash-in rate and the peak enhancement show some similarities. You can see this area down here, which is showing really poor peak enhancement, but there are some differences within the areas. So these parametric images are quite nice in that they allow you to very quickly and easily separate out those differences. Again, to reinforce, all of these data are widely and easily exportable from the system. The parametric images as TIFF images, so you can compile them into a nice image for a paper or a presentation such as this one, and then the actual numerical data exportable as a flat text file that you can take into Excel. So lots of papers out there. I've put examples of 3 different papers in here that have to do with nonlinear contrast imaging on our system. This first one here, this one at the top, is one that actually mentions VevoCQ and actually shows some parametric images. I believe it's in the supplemental data. The other 2 are nice examples of some of the work that's being done in nonlinear contrast imaging with the Vivo 2100 system. If you look on our website, on the customer portion of our website, there is, if you don't have access, you can just request an access. It's free and open to anybody to request. Have a look on there. There is a searchable database of papers that have been published that reference or use the system. You can search for terms like contrast plus kidney or tumor or cancer, mix the terms together and look for different areas that you might be interested in. So if for anybody who has a system or is thinking about getting the system, I really recommend having a look at that on our website, on the customer portion of our website. It's a great resource to really see the types of work out there being done with the system. So we'll move into a little bit of details now for people who are actually using the system. I just wanted to give you guys some pointers and hints about image acquisition. Being a powerful, sensitive post-analysis package, VevoCQ is really beholden to the type of data that you feed it. You really need to feed it good quality images if you want to get good quality data out of it. So some things to remember it has to be nonlinear contrast, so that means either using the MS200 or 250 and capturing images in nonlinear contrast mode. Very key. Things like the image size, so the frame rate, the image width, and the frame buffer size, these to varying degrees together and apart will control the amount of data that you have. VevoCQ requests that you have a fairly long clip, 20 seconds or greater, which will give time in perfusion kinetics for the microbubbles that have been injected to come up to close to a constant or peak level of a circulating bubble signal. And that really helps you improve your analysis. You can change things like the frame buffer size by adjusting the frame rate and adjusting the image width. The frame rate, there's actually a controller knob on the system in I believe it's in the center left, or middle left, sorry, I should say, not center, where you can dial up or dial down the frame rate. adjusting the image width, so the actual width of the image. We have an example here of the mode settings panel from the 2100, and this actually shows a width of 19 millimeters. You can stretch that out or clip it in to really focus in on the area that you're interested in. The image width will affect the frame rate, as also will the number of focal zones. Some of the current version of our software, we've changed it so that you go to one focal zone in nonlinear contrast, which is the way it should kind of be. The earlier versions, you could have more than one focal zone. Hopefully, you've been adjusting that a little bit as you've been imaging. One other point to add with the frame buffer size, which you can have a nice effect on, is if you go to the preferences window on your system, just hit the prefs key, you'll notice frame buffer sizes for the different imaging modes. And in the contrast modes, nonlinear and linear, you can actually use an extended buffer. This extended buffer adds significantly to the amount of data that you're capturing, so you have to bear that in mind. It may mean more processing, but what it does do is give you a really long run of time to allow for data to really accrue in the buffer, and for hopefully the bubbles that you've injected to come up to a constant circulating level of perfusion. One other thing to mention is the TGC sliders. These are the time gain correction sliders, TGC on the 2100. These are the black rubber sliders that are up in the far upper right corner of the keyboard. Changing the TGCs allows you to correct the gain in different parts of the image, all the way from the top of the image down through the bottom. You really want to try for contrast studies to keep these consistent through your study, so that you're imaging your animals or your groups of animals with consistent settings of gain. So there's a couple of different ways of doing that. One is to just set the sliders to the little catch in the middle. If you slide those sliders back and forth in their grooves, there's a little catch in the middle where they'll sit. So if you set them all in the middle, that's a nice home position. The other one is if you go to the Image Process tab on the system, there's a selection there where you can actually save the TGC settings. So if you can't have them set all in the home center position, if you need to have them, say, dialed up or dialed down in the upper or lower portions of the image, you can set that and then save that setting. That will allow you to recall that setting and then lock a recalled setting in place. So then that allows you to keep those consistent throughout your studies. So just to sort of tie things up at the end of the presentation part here, we've gone over the basics of VevoCQ, about sort of what it is, what it's capable of doing. It's a very easy to use advanced post-processing analysis tool, and this allows you to really dig down really deeply into the perfusion kinetics of the types of tissues and animals and data that you're really interested in looking at, much further above and beyond the standard quantification tools that are available on the 2100. There's a number of different modes available, perfusion, targeted, and destruction reperfusion. In the demonstration portion, I'll give some examples of standard bolus perfusion and destruction reperfusion. And we'll open the floor up to a questions interlude. If anybody has anything that they would like to know, please go ahead.
Okay, thanks, Stephen. I don't see any questions at this point. In the next 30 seconds or so, if you do think of anything, please ask. Stephen's just going to quickly switch over to the Vivo 2100 software and show you an interactive session here using the VevoCQ in action.
Okay, great. Yes, and as Andrew said, please, if you have any questions along the way, feel free to send them on through. So I've got some data here that has been done previously. We have some subcutaneous tumor, targeted and untargeted. What I'll actually do is go into this first image and just show you how things look typically from your normal imaging modes when whether you're on the system or on the desktop. We have a nice bit of perfusion here coming into this tumor. Kind of poor perfusion down here and better perfusion here. Those of you who have done nonlinear contrast on the 2100, you'll be familiar with this. Bringing up the measurement panel, and going to contrast region of interest. So I'll just very quickly draw a contrast region of interest that covers the whole tumor, and we'll go to the region graph very quickly. So this gives a nice example of the basic analysis that's available on the system. Now this is standard. This comes with nonlinear contrast mode, and with this you're able to look at an area here, look at a region, and get peak enhancement and time to peak for the perfusion that's in this region of interest. We can draw multiple regions of interest on this tumor, and then these data are able to be exported out. So we can export the constituent components of this chart out to an Excel file and actually rebuild this and do some analysis offline using the hard data. If we want to go into VevoCQ, though, and take our data analysis that much further, there's a couple of different ways of doing it. We can go on the measurement panel again, up here in the upper left, there's a button for VevoCQ, or if we go back to the study browser, I'll actually show you there's a VevoCQ button here. Wherever you have a nonlinear contrast image selected, VevoCQ button will be available if you have it on the system. This area here from the study browser is nice to go into because you can actually select multiple images. So we've highlighted these two images here, and we'll actually put those into VevoCQ at the same time. So these will go in, and we'll have some data here that we can recall. This is a good point about VevoCQ. It's very nice that you can actually recall your saved data. So when you go ahead and do your analyses, you're able to go back in and pull those back up. So this one here is loading from the saved data right now. We can see the regions of interest that have already been drawn here, and the system is busily loading the clip here. While it's doing that, I'll actually jump over to this one here, which I chose to not load the data from. And we can start drawing regions of interest. So this will be sort of the main screen that you would go into for VevoCQ. The first time you ever put an image in, you'll come in here. You can draw a region of interest. Once you've set that region of interest, you can move those points around, make some adjustments. I'll go through drawing the region of interest again here in another area. So I'm just picking another region and clicking points here with my mouse as I go. And then when I get close to the origin where I want to close it off, oops, it's jumping around here on me. Let's try that again. It just jumped over to the other one because it was finished analyzing right in the middle of everything here. Here we go. Third time's the charm. So we'll draw a region of interest around here. We'll double click to close it off. Again, we can make some adjustments here, and once we're happy, we can quantify. Now, this one is a short clip. This is complaining because at 19.5 seconds, it's half a second too short for the suggested minimum of 20 seconds, but you have a choice to click through that. So we'll click through that. Now, while this clip is loading and quantifying, we'll jump back over here to the saved data that we recalled. So this is saved data that was recalled. Again, it's a bolus model, a bolus injection. And I believe this is actually the same tumor image that we saw during the presentation. So we can see here the regions of interest. There are 4 here that were drawn. These are adjustable here in this image too. We can actually bring the points back up, make some adjustments if we want, and there we go. Go back here, make some adjustments, and see things change. Now, we'll leave these regions of interest alone. I wanted to point out down here in the lower left, we have the curves. So these are the curves that the system has plotted out for what it sees as perfusion in these regions of interest. Again, color-coded, so you can see total tumor in green, and then purple and yellow for the other areas. If we come over here to the right side of the screen, we can see a table here, and let's choose from the drop-down list here. We have a wide range of metrics that are available. So we talked a little bit about peak enhancement and time to peak, but we can also look at things like wash-in rate. As soon as we choose one of these, we get the parametric image up here in the upper right, and then in the lower right, we actually get the numbers, the arbitrary units and decibels that are tied to the wash-in rate for those particular areas. And if we change these to something like the rise time, you can see the parametric image changes quite drastically, and again, we have a different set of numbers down here. Now, some of these parametric images are static. Some of these ones, say for peak enhancement, wash-in rate, rise time, area under the curve, things like that. They are static versus the other two. Let's see, we'll look at the fitted signal and the linearized signal. These are actually dynamic and they will change with frames. So you can actually go through, so we're playing the clip here now, frame by frame by frame, and look at how the signal, the linearized signal, the fitted signal, changes in those regions of interest. And we loop back around here, you'll see the signal disappear, and then it comes rushing in as the contrast agent comes rushing in. So again, these are all exportable. Again, you can export the entire screen as a TIFF to put into a presentation or a paper, a series of parametric images, and then all of these data that are here, the standard data, and also some of these other data that are based on extrapolated data, extrapolated beyond the end of the curve. These are all, again, exportable. So if we jump over to the other one, I kind of clicked through it quickly because I wanted to show the area. It actually came up with a screen, with a question screen, once it had done the quantification, asking if the point that VevoCQ had chosen for the arrival of the contrast agent was acceptable. And I just clicked through it as saying yes, that it was. That's usually what I do. You can adjust it, but most of the time I've found in my experience that the algorithm, the detection algorithm, is more sensitive and more appropriate, so it's better for it to actually choose the arrival time. So we can see a different profile here. Again, we've got some differences in the total region here, the total tumor, and then the more highly perfused region that was up here in the upper side, in the upper left side of the tumor. And again, we're getting nice images here. Now we can change these tags. These are just tagged as ROI1. This should be tagged as ROI2. This is just a holdover from a previous analysis. We can actually change this to, say, perfused area, and change this other tag to total tumor. So you can adjust those as you go. Make changes as you go to mark on your data the particular areas that you're interested in looking at. So I'll close out of this temporarily, and we'll jump back in with another example of destruction and perfusion. I just wanted to point out here that when I click close, it sees that I have two clips open, and it wants me to make sure that I want to close all of them. I'm going to say no, that I don't want to save the results because they're already saved. And while we're here, I'll just point out that because we did a VevoCQ analysis, this is marked on here on the study browser that VevoCQ analysis has been done, and that's something that you can come back to and pull up again going forward. So I'll actually cancel on this one because I want to show you the difference here in the main screen. Now, in the original first ones that we pulled in, bolus was already selected, and we can actually select bolus if we want to. This type of image has a destruction pulse, a burst pulse here. VevoCQ actually sees that and decides, oh, you must be using a destruction, reperfusion, replenishment model, and chooses that for us and eliminates the frames before the burst pulse was applied, and then also during the burst pulse, and then wants us to focus on just the frames after the destruction pulse. So we'll do that. We'll click quantify. Again, it's complaining that it has a short clip. That's okay. Short clip is maybe better for us because it will load a little faster. So it's loading the clip in now, and it's going to take a few seconds to quantify the perfusion again in the region of interest. But once we're into the analysis screen now, again, we're seeing up here in the middle upper portion of the screen how it's marked as replenishment model. And if we click the drop down here now, we've got some different metrics here that we didn't have before. We have things like relative blood volume and relative blood flow that are available in the replenishment, the destruction reperfusion model versus the bolus model. Now, if we were to change it to the targeted model, again, we would also have some different selections there, including differential target enhancement. So it just the software just gives you the flexibility to look again at different models. So again, here we've got the same analysis that we had before in the previous screen. We have the replenishment plotted. So you have to bear in mind again that what's being plotted here is actually the area after the destruction pulse was applied. So this is the replenishment curve, and we can look through the different metrics mean transit time of 0.8 seconds, relative blood volume in arbitrary units in decibels as well. And of course, as we're clicking through those, the parametric image is changing in kind to reflect the different metrics that we're looking at. So I'll close out of this. I'll click No to save, and we'll actually jump back to the end of the presentation. And I think that's pretty much it for the software demonstration portion. If anybody has anything extra they would like to add, we can do it now, and we'll open the floor up to questions if there is any available.
Okay, Stephen, great. That's fantastic. I'll just give people a few minutes to type in any questions. Again, just a reminder, if you can use the chat window, if you do have any questions, we can certainly take them through the chat, and then Stephen can try to give us a response.
Okay, so excuse me, we have a question here. Thanks, Andrew, about the experimental model. So VevoCQ will recognize whether it was a bolus or a destruction reperfusion for you, just based on whether or not there is a burst pulse in the data stream within the image. In terms of choosing the model, that's more up to experimental design, whether you want to do a bolus injection, a single bolus injection, and watch the wash in, or do a destruction reperfusion and destroy the bubbles in the frame that you're looking at, and watch for the reperfusion to come in. There are lots of arguments for both and against both for a variety of differences. So if you're thinking of designing an experiment to use nonlinear contrast, I encourage you to look into the different sort of approaches that are out there to use. VevoCQ fundamentally doesn't really care, I guess, is the best thing to say, which one you use, as long as you have a good image, and as long as you're confident in your injection or in the destruction reperfusion in the area that you're looking in, that it is reflective of the tissue that you want to look at.
Okay, Stephen, thanks. That's great. Stephen, just another question about short cine loop lengths. You mentioned that if the cine loop is less than 20 seconds, the software prompts you. The question here from one of the listeners here today is, why do you recommend 20 seconds, and what might happen if it is shorter?
Okay, thanks. So the idea behind the 20 seconds is that generally for most tissues, a 20-second stream of images gives time for the microbubbles to perfuse into the area of interest and to come up to a constant circulating level of perfusion. So this allows, if you think back to the curves that we showed, this allows you to reach a peak level of circulation. So if we look on the screen here, we've hit this peak in the purple here in this highly perfused region, and then the signal drops off a little bit and then kind of stays at a constant circulating level. This is a sort of a normal, kind of a standard model of perfusion. For some of the analyses that you're doing, things like rise time or mean transit time, the software is anticipating being able to capture this rise, this peak plateau level, and then this drop off. If a clip is too short, say 5, 10, 15 seconds, there may not be enough time, depending on the tissue that you're looking at, for the level of signal to come up to that constant, that peak level before tailing off. So some of those models, some of those parts of the modeling algorithms don't really apply. So the software will complain. It'll say you should have something longer. And then in some cases, if it's too short, you'll also see an exclamation point and a value of NA, and that's because either the clip was too short or the area was too slowly perfused for the tissues to actually get up to a full level of circulating signal for it to be able to apply those models to. So playing around with the frame buffer, playing around with the size of the image and things like that will allow you to stretch that time out and hopefully give you enough time to capture all of the data that you need for the signal to get where the software wants it to be.
Okay, that's great, Stephen. I hope that's helpful for a lot of people to understand how that bolus model is actually working, and giving enough time to let it really do its job. There's a question here just about VevoCQ, and has it been tried in animals other than mice?
So yes, we do image a fairly wide range of different types of animals on the 2100 system. Probably the bulk majority are mice, quite a lot of rat imaging, and then lots of other animals too. I've done nonlinear contrast imaging on both mice and rats. They work equally well. Typically on a mouse we do a bolus injection of 50 microliters, usually around 4 times that for a rat. The quality of the data is quite good, and works just fine with no problems in VevoCQ. I can imagine that contrast data on other animal models too, say on rabbits or guinea pigs or other types of animals, would probably work just as well too. It's with something like this being a post-analysis, post-processing package, it's more about the quality of the data and the type of data rather than the animal that it's from. As long as you have good quality images and good quality nonlinear contrast data, you'll be able to do some analyses on it with VevoCQ.
Okay, great. That's fantastic. At this point, I don't see any further questions. We're running just a couple of minutes ahead of schedule, so perhaps just to conclude, I wanted to just give everyone a few reminders. First of all, I wanted to bring to your attention a promotion that VisualSonics is currently offering. Our micromarker contrast agents are being offered at 50% off until the end of June, so that's until June 30th. We are also offering trial licenses of VevoCQ and the nonlinear contrast mode software. So if that is something that is of interest to you, that's a 6-month promotion, started in January, runs to the end of June. Please contact us at info@visualsonics.com. Your email will be directed to either myself or to Stephen. We can follow up with any other questions you might have with CQ or nonlinear contrast in general. Finally, we will be sending out an email after today's session with a link to a recording so that you can go back and review the useful information that we had here today. And also there'll be a link to a short survey that we'd really appreciate you taking the time to fill out to get some feedback both on the content of the webinar itself, as well as any other some other imaging type feedback we'd love to get from you. And finally, we do offer webinars, as you can see here, visualsonics.com/webinars. So please join us in the future. Appreciate you taking the time out of your busy schedules today to join us, and I hope that you've learned something more about VevoCQ that can help you in your research. So thanks to Stephen. We'll sign off for now. Take care.